All right, now we're going to be looking at the mammalian embryo. The mammalian embryo, like the Amphioxus, starts out as a microlecithal egg, very small cell, tiny little cell, and it will undergo a complete holoblastic cleavage, unlike the frog and the chicken egg that have yolks to deal with. But the developmental process in the mammal is not very much like what we saw in Amphioxus. It has some major differences. We go from I should draw that bigger, but anyway, we go from a one-cell stage to a two-cell stage to a four-cell stage, and here the cells are kind of offset to one another, kind of a tetrahedral kind of arrangement. Then you go on to the 8-cell stage and so on. So you have this three-dimensional sphere of dividing cells, and eventually you arrive at the morula stage of development, that berry-like stage. So this is the zygote up here. This is 2-cell, this is 4-cell, this is 8. This is probably more than 16, but anyway, we're at a morula stage. At some point, the synchronous division of the cells breaks down, and you no longer have the even number of cells that you begin with in your early cleavage stages. But the next event after the morula is the formation of the blastula stage again, and here's where things get a little bit different from the other groups that we looked at. So we have this ring of cells that forms an outer layer on the developing embryo, but inside there's what's called an inner cell mass, a group of cells inside here that will divide to form most of the structures of the embryo. There's still a blastocele cavity in here. And then we have this inner cell mass. Those cells that form this ring around the outside, and we're going to color them green in the pictures that follow, but anyway, these cells around the outside, we're going to call these trophoblasts, T-R-O-P-H-O blasts. Trophoblasts, or feeding cells is basically what that means, trophoblast cells. So here we are looking at the mammalian embryo developing. We have the outer cells that are called trophoblasts. The inner cell mass is now changed somewhat. We've got a cavity developing inside that inner cell mass, and that is going to be the amniotic cavity. There's a layer of cells here that will form the ectoderm of the embryonic disc. Very much like what we saw on the surface of the chicken yolk, how that embryo was developing in the bird embryo. The lower layer of cells across here in this embryonic disc, this is going to be the endoderm layer. So we have these cells that are considered all ectoderm up here, including the embryonic disc and those above the amniotic cavity out there. So everything in blue is ectoderm. So I'll go ahead and label this. I'll just put in parentheses here, ectoderm. All that in blue. We still have on the inside here the blastocele cavity. The blastocele cavity on the inside, trophoblast on the outside. So the next thing that will happen with this is these cells will keep proliferating, getting smaller and smaller with each division, and these cells of the endoderm will spread around the cavity of the blastocele, which is different from what we saw in the other kinds of embryos we've looked at so far. So next we'll draw the next stage over here. So we still have many of the same things. We have the outer green layer here that represents trophoblast cells around the outside. We have this cavity that was blastocele cavity. It's eventually going to be identified as our yolk sac cavity. This space above is amniotic cavity. There's a layer of surface ectoderm layer across here, and then below that a layer of endoderm. And our embryo proper, the whole body of the embryo, is going to form from just this center part of that embryonic disc that we have forming across here. The cells of the lower layer are going to move in around here to close in that whole space to make that yolk sac cavity. And this is essentially similar to the epiblast-hypoblast layer we saw in the chicken embryo. The ectoderm and the endoderm below it is similar to epiblast and hypoblast layers there. So in that area, we're going to see the formation of a primitive streak, and eventually the whole body will form from that area right there. For a little while, we're not going to pay much attention what happens in that body region. We're going to make reference to it now and then, but we're not going to show its development. We're going to concern ourselves with what's happening to the extra embryonic tissues. Now one more picture down here. So our next stage in the progression, the trophoblast layer is still on the outside in green, the endodermal tissues indicated in blue. The ectoderm in blue, the endoderm in orange. In this case, we don't see any mesoderm shown anywhere on this yet, and that's going to be one of the next things we see in this illustration here. Off of the outside of the developing cells of the ectodermal bubble and the endodermal bubble we have growing here, we get some more cells that stem from that and expand out in between the trophoblast layer and the other inner cells in here. This will eventually surround all of the inner structures of the developing embryo. So we have the formation of extraembryonic mesoderm, Now the trophoblast, the cells keep dividing here, and some of these cells on the outside begin to form a syncytial mass, and we're going to put little dots in the syncytial regions of trophoblasts. So we now have regions with syncytial trophoblast. SYN, CYTIO trophoblasts. Syncytial trophoblasts. The others are still cellular, individual cells and individual nuclei wrapped in their membranes, and those are considered cytotrophoblasts. Cytotrophoblasts, or cellular trophoblasts. These out here are syncytial. That's a wall that breaks down or separates the cells, breaks down, and or the membrane that separates the cells breaks down, so that all the nuclei are gathered together in one mass, and that will continue to form all the way around the embryo. So there'll be a layer of syncytial trophoblasts around the outside, and then cytotrophoblasts closer to the embryo on the inside. I mentioned that in the red, this is considered extra embryonic mesoderm. Extra embryonic mesoderm. This down here is now going to be our yolk sac. Here, our amniotic cavity up here, and as we said, our embryo proper will be developing. I'll just kind of put brackets around it. It'll be forming right in that zone right there. That's where our embryo will form. In the next stage of development, we're going to show this extra embryonic mesoderm expanded around here, around all of the inner structures, around the endoderm and ectoderm portions, and within that extra embryonic mesoderm, a space starts to form. So we're going to have a cavity around the outside of that, a cavity with trophoblasts and mesoderm to the outside, and then mesoderm and endoderm, or mesoderm and ectoderm to the inside of that space. All right, we have on around the outside our syncytiotrophoblast layer with the dots in it. Then we have our cytotrophoblast layer, the next ring in from that, divided up into hundreds and hundreds of cells, thousands really by this time. To the inside, I've already drawn in my ectodermal portion, the amniotic cavity and yolk sac cavity, and where the embryo forms right in here. And now, surrounding that, there's a layer of mesoderm. Kind of scratch that in there. A layer of mesoderm to the outside of the ectoderm there, and a layer of mesoderm to the outside of this part here. And then near the tail end of the developing body, there's a connection of this mesoderm to a layer of mesoderm that's out toward the outside of the body. Outside of the embryonic structure, up against the trophoblasts around the outside. So this part right here, where this mesoderm that's associated with the inner structures connects to the mesoderm that's next to the trophoblast layer out here, this is simply referred to as a mesodermal stalk. That part right there, that's a mesodermal stalk. The three layers we have here, that layer of mesoderm here, the trophoblast, cytotrophoblast, and syncytiotrophoblast together, all three of those together makes up what we call the chorion, or chorionic membrane. Chorionic membrane. Inside of that, that space there can be either called the chorionic cavity, or extra embryonic coelom. Either name would be fine for that space there. The next layer in here, this endoderm and that mesoderm together make up the yolk sac membrane. The mesoderm and the ectoderm up here make up the amniotic membrane. So we still have our amniotic cavity, we still have our yolk sac cavity, and we still have our two-layered embryonic disc region right there. And it's in that layer right there that we'll get the primitive streak to form. Primitive streak will be folding in to make a new layer of mesoderm, a different mesoderm from this extra embryonic mesoderm that we've already seen here. So the next events that occur that are vital to the formation of the body are occurring right in here. We'll maintain this condition of having the syncytiotrophoblasts, cytotrophoblasts, and all that together for a while. And as the embryo starts to grow inside the amniotic cavity here, as it grows up and into that amniotic cavity, the body will grow and eventually push the layers of mesoderm around the amnion and the yolk sac. They will be pushed out against the chorionic mesoderm on the outside. So we have chorionic mesoderm, we have yolk sac mesoderm, we have amniotic mesoderm, and we have a mesodermal stalk connecting them across here. Okay, now we're back to a familiar drawing. We saw this with the chicken embryo. We have the head process growing here, the tail process going back there. We've seen the formation of a neural tube and a brain developed from the front part and a spinal cord developing back here. From the gut, the orange layer here, we have foregut coming up here to the mouth area, and then the hindgut developing back here to the cloaca. And then off of the hindgut, we have this tag right here, which will be the allantois. And we still have this connection to the yolk down here, the yolk sac, anyhow. So I'm going to put some mesoderm on this now. Remember, there is a mesoderm, need red for mesoderm, mesoderm down the middle underneath of the spinal cord right here. That is the notochord, right? The notochord down through here. There's a connecting mesodermal stalk off of this end of the embryo, and that mesoderm comes up and then wraps around the outside of the amniotic membrane like this, all the way around the embryo here, and then it comes back in around the outside of the yolk sac here, and then connects back to that outer wall. And this outer part is going to be the chorionic mesoderm around the outside, around here. So around the outside of the mesoderm here, by the way, the mesoderm has a certain thickness we need to put in there, so we'll put another layer to represent that thickness here. And near that body stalk back here, that mesoderm begins to grow out into the cytotrophoblasts and syncytiotrophoblasts as what we call chorionic villi. Chorionic villi. So adjacent to that is where we have our cytotrophoblast layer. And then outside of that will be our syncytiotrophoblasts. So another layer of syncytial cells. Remember, they're not separated by cell membranes. All their nuclei are together. So we have this outer layer of syncytiotrophoblasts and cytotrophoblasts and mesoderm. So in this picture, there's our chorionic membrane. This is a chorionic villus. Chorionic membrane, then we have our yolk sac membrane here. drawn our allantois in this case it's growing out into this mesodermal stalk, so it's surrounded by mesoderm but not in a single layer. So there's the allantoic sac going out there. Some foregut. Bringing out a black marker here. Hindgut. This is midgut. Here's our brain. Spinal cord. There, we've got a notochord going through there. Amniotic membrane. So we won't label anything else here. We've got the amniotic cavity, obviously this space here. We've got the yolk sac cavity down here. And as I said before, this embryo will continue to grow, and as it gets bigger and bigger, it's going to push that amniotic cavity more up against the chorionic cavity. This yolk sac is not going to grow any further. It's going to be taken up into the body stalk, and I don't have the body stalk identified there, so it'd be good to identify the body stalk. I need more colors, but anyway, right around here would be the body stalk, that connection between the embryo and all this other extra embryonic tissue right here. There's our body stalk. Eventually we'll recognize that as an umbilical cord, Now, notice how we have this connecting mesodermal stalk back here in the back still. We can label that, I guess, still mesodermal stalk. Those blood vessels that come off to the allantois, remember our heart is here, we have our aortic arches that go around the pharynx, a dorsal aorta above the gut. There are blood vessels that go to the yolk. Here, the vitelline arteries, vitelline veins go back to the heart. And then back here, there were allantoic arteries that go off to the allantoic sac. In a mammal, in a placental mammal, those vessels continue right on out through the mesodermal stalk and spread out into these chorionic villi. And that's where you have contact between maternal tissues and the embryo tissues. Right there, that nutrient delivery from mother to developing embryo occurs out here. This is the formation of the placenta, right here. So remember, your allantoic vessels become your umbilical vessels. There are two umbilical arteries, or allantoic arteries, that reach out across that mesodermal stalk to the chorionic villi. And there's one vein that comes back the other way, then enters into the back of the heart. Over here, one umbilical vein, two umbilical arteries. Two vitelline arteries and two vitelline veins to the yolk sac. But remember, that yolk sac eventually just gets taken up into the umbilical cord as the umbilical cord forms in a human embryo in most mammals, placental mammals. Okay, just a little further along, I drew the embryo larger in this case. We've expanded our chorionic villi here in the placenta region that we're forming. This is the head right here, large head on a young embryo. This would be the rest of the body and the very end of the body way back there. We've got the brain, we've got the notochord in red, and in orange we have endodermal tissue, so this is foregut. This is hindgut, and right there's the allantois coming out through the umbilical cord. This is the umbilical cord right here. allantois comes out through there, and the connection to the yolk sac. So this is all that's left of our yolk sac. It didn't grow much, so as everything else grows around it, it gets smaller and smaller and is eventually taken up into that umbilical cord as just a remnant of what it used to be. But the extra embryonic mesoderm that we had before that was formed a single layer to the outside of the amnion and this, on the yolk sac and then around the chorion, it now occupies this space with no chorionic cavity or extra embryonic coelom space left in there. So we have this thin layer of mesoderm between the ectoderm of the amniotic cavity and the trophoblasts out here. But all of this space here is taken up by mesoderm that reaches out into the chorionic villi. And my marker's not the best for filling up this space here, but it comes in around the yolk sac and around the allantois, in the umbilicus or body stalk area. Fills up all this space around the yolk sac. And extends out into the chorionic villi. So it's in this mesoderm that I'm drawing in right here that we're going to find those blood vessels, the branches of the umbilical arteries and veins that carry the blood. And then we have back to this narrow space between the amniotic ectoderm and the trophoblast layers. So the extra embryonic coelom or chorionic cavity is gone because of the growth of the embryo here and the expansion of the amniotic cavity there. So that pushes all those membranes out against the outside. Recall that our heart is located like right here underneath the pharynx, sends the blood out through vessels that go either to the yolk sac, which are no longer that important, or to the allantois, and those are very important because those arteries that go to the allantois branch out right here to go through all the umbilical structures, all these chorionic villi of the umbilicus here. And then veins return the blood the other way. If there's a single umbilical vein going through the umbilical cord to the back of the heart.